Thank you all for coming to our webinar. I would like to give you a short overview of some exciting data that we've been generating in our lab in collaboration with a number of colleagues to try to use mice to unravel the mysteries of the COVID-19 disease epidemic that we are all so aware of. I'll just say a few words in support of viruses. We think about viruses as real pathogens on a scourge on our planet, but it's been said that if viruses disappeared altogether, we would live for another day, and that would be about it. It's an interesting fact that viruses actually control a lot of the infectious agents that would otherwise overwhelm us and other living organisms on the planet. And with regards to human, they've actually contributed to a number of aspects of our own evolution, including our expansion out of Africa. They have definitely influenced our immune system through the balance of the microbiome that they can control. They move genes around between people. One amazing example of a virus function that has lived on, co-opted from viruses into our own biology is the evolution of the mammalian placenta, without which we would not be where we are today. So although we spend most of our time trying to figure out how to get rid of this virus, I just like to say that they're here to stay and that they are actually very, very interesting to study, and even more interesting to develop cures for. So I don't have to spend much time on my introduction because both Caitlin and Jim did such a fabulous job. Here's another picture of this little devil. There's a funny way of describing viruses, which is that they are essentially a bunch of trouble wrapped up in protein. And the trouble is what you see on the inside, that slinky looking thing, which is their genetic material, and then the spike sticking out, which is where we know the virus uses the relationship with its spike to bind to the ACE2 receptor, which Caitlin described so very well, and that allows the virus to get into a human cell. And then it co-opts the human cell's own machinery to replicate itself and produce many more viruses that then can go out and infect other tissues. And the tissues that get infected are really varied. And I think this is one of the major reasons why the SARS-CoV-2 virus has become such a real problem. When you consider all of these tissues, each one of which has the ACE2 receptor on its surface of the cells, there's no small area of your body that probably isn't at some level susceptible to infection. But the mystery that I mentioned is really the variation in the clinical output of the viral infections that we know about. So we already are aware of the incredible variation in our own response to the virus and the outcomes that the disease causes, anything from a minor cold or full recovery, right through to ending up in the ICU. And we also know that comorbidities are very, very important. When we first had the pandemic sort of surround us, it seemed that younger, healthier people were much less susceptible and that the virus selected out older people with chronic conditions. And that's still true, but it turns out that now with some of the new variants, younger people are more frequently getting sick. And then comes the whole problem of long COVID, which is a slang for long-standing effects that go on beyond the initial infection, such as all sorts of problems with your immune system, headaches, and other debilitating diseases. So what is it that makes us so different one to another? And how could we predict who would get really sick and who wouldn't? It turns out that the disease severity is really caused not by the virus itself, but by your own host qualifications. And of course, being a genetics institute at the Jackson Laboratory, we're particularly interested in whether genetic differences would make some people more or less susceptible to the SARS-CoV-2 virus and its causes of disease. So given that mice are a wonderful tool for understanding genetic differences, we immediately turned to the mouse. And of course, as you heard from Caitlin, one mouse is not enough. Of course, there is an enormous amount of power that comes from looking at inbred mice, but you don't have to limit yourself to one. And as anybody who's perused our catalog knows, we have not one, but 11,000 different mice that you can choose from. So in theory, that would allow us to study the mechanisms of SARS-CoV-2 viral infection, look at the complications, but not just in one background, and then follow up long-term problems with disease progression, develop new diagnostic approaches, and test new therapeutics, and do that in a way that is increasingly relevant to the human condition where we are also genetically variable. So as you heard, mice don't get COVID-19, and the reason for that, as you also heard from Caitlin, is that the mouse ACE2 gene has very profound differences from the human, and therefore it does not bond effectively to the spike protein of the virus. And that was really the rationale behind making a transgenic mouse, the K18 human ACE2 transgenic mouse, which you heard about before, which has an extraordinarily broad use in modeling vaccines and antiviral development, but does have the problem of being almost too susceptible to the viral infection, resulting, as you heard, in a very rapid progression to death. And here again is the same sort of data, just from a different lab, showing how weight loss in these animals is profound and is somewhat dependent upon viral titer. So that red line down here is the most severe from a higher viral titer. This line here, the pink line, is a less strong viral titer, and that, of course, the green line is the uninfected animal. And as you can see on the right, there really is a profound drop-off in survival just about under a week. So this really limits the amount of pathology you can study in these animals. But we were also aware of the fact that there may be something about the Black6 background which is contributing to these very steep survival curves. And really the concept here is pretty general. If you use one mouse to study a whole bunch of people, you're really only probably going to be modeling one out of many of those people. Whereas if you study a number of mice, you're much more likely to be able to pick up differences that are relevant to a larger population. And of course, as we have known, the differences in the disease itself really almost requires this kind of approach. So here at the Jackson Laboratory, my lab decided to try this trick out by essentially crossing that K18 ACE2 model with the humanized ACE2 transgene with a whole bunch of genetically diverse mice. And in this case, for the aficionados, what we used were the founder mice for the collaborative cross, 8 different inbred strains, which we then crossed into, we crossed the transgene into it, so that each mouse that comes out of this cross, the progeny, all are half Black6 plus the transgene and half some other genome. Of course, all mixed up, not like this, but this is just a cartoon. And the concept there is that if we then infect these animals with SARS-CoV-2, we might pick up differences that could mitigate this very, very severe phenotype in the original K18 ACE2 model and give us a much more nuanced look at how mice might model human disease. Unfortunately, at the Jackson Laboratory, well, fortunately for all of you who use our mice, we do not have infectious agents on campus. But unfortunately for this project, it was a little bit of a problem until we got in touch with the National Institutes of Allergy and Infectious Diseases Rocky Mountain Laboratory outpost in Montana, where many even more severe viruses are studied. And just for those of you who have any familiarity with PPE, this is the level of PPE you've got to use in the Rocky Mountain labs where they're studying things like Ebola and Zika. So it is not for the faint of heart. Luckily, we had some close colleagues who got very interested in this project and agreed to take all those mice, all those F1 mice crossed from the Black 6 transgenic mouse into the new strains and test them for CoV-2 infectious signals and signs. And I'm just going to show you a little bit of data because it's just too much. It'll overwhelm you. But this is all the take-home message is that simply introducing half of the genome from another strain really radically changes the way these mice respond to the virus. So here is just a little sort of sampler for you. On the left, you see the cross to PWK, which is a wild-derived strain. And look at that. The actual survival curves are almost flat because these animals simply don't seem to get sick. Then here is the B6, the one that we are all looking at and using, and again, another canonical drop-off of survival. And we got some with sex bias. In this case, we're looking at the cross with NOD, and it seems that the males are susceptible, but the females are not. And we had others that actually did all sorts of other combinations, which I'll just go over very briefly. When we looked at the pathology, we were it was very clear that these animals were getting infected, no question about it. On the right, you see viral infection as red, and on the left, you can see the damage done to the lung. And just for those of you who are interested in brain, there was a very interesting pattern in this particular cross where you see on the left that viruses are getting into all parts of the brain, shown here in red. And the hindbrain, for reference, is over on to the left. So it's as if the mouse is facing to the right. But in the pathology analysis, it looked like only hindbrain was being affected by the viral infection. And just by chance, that's where your sense of taste sits. So we all know that that's one of the things you lose when you get COVID-19. So obviously, there's tons and tons of interesting data to peruse here. And just to give you a feeling for the differences which you don't need to read here, you can see that some of the features are male specific, others are female specific, and down at the bottom, that PWK mouse just doesn't have any symptoms at all. Well, what's really going on here? Obviously, we're still working hard on these animals, but I'll just give you a little bit of information. I should say that all the work that you are seeing is a collaboration with Dr. Sonia Best and Shelley Robertson out at the Rocky Mountain Labs. Anyway, what you're seeing here is a titer of viruses, either on the left in the circles in females and on the right in squares in males. And here's that cross I just told you about, PWK times Black 6 with the transgene. And these are the mice that walk away. And you can pretty much see why. In the lung at the top, you can see that after 3 days, there's a significant titer of virus, which is completely eliminated after another 3 days. So by 6 days, these animals have cleared the virus entirely in both males and females. That's just shown with that little blue arrow right here. And they actually don't appear to have any infection in the brain, probably because by the time you get out to 6 days, there's no virus to spread to anywhere. In contrast, this is the K18 animal. You can see that virus persists right up until death in the lung and starts to accumulate in the brains, in this case of males. So it's clear to us that there are genetic differences between these animals and these animals that can completely account for the way in which this mouse combination survives and actually gets rid of the virus altogether. Anyway, there's obviously an enormous opportunity here. We have looked at 8 different combinations, and we have gotten 8 different phenotypes. And of course, with the number of different kinds of mice we have available, this could go on forever. And we're obviously very interested in pursuing more crosses with more different combinations and maybe having more than just two parents by crossing some of the offspring to see just how detailed and nuanced we can get these models. Ultimately, what we would like to do is to be able to have a model for every human condition so that then we can be very much more precise in our drug discovery using only the model that actually makes sense for that condition. And obviously, we're very excited about this project to the extent that we're about to make these mice available to the general public and to you for further analysis, because obviously we can't do it all ourselves. However, as you heard, there are many, many more components to the viral life cycle in cells that may need humanization in the mouse. And so we are undergoing a number of experiments within the Jackson Laboratory to humanize as many different components of the viral life cycle in human cells as possible. So introducing humanized versions of these different genes and even a humanized immune system. Now, ultimately, the other thing we would really love to do is to take the mice that can survive and let them go out for longer, because we've only looked for about 3 weeks so far. It's early days and we're trying to hurry up and get this out. And ideally, what we would really love to do is to see whether the different mice end up on the long road to long COVID at different places and get different symptoms. And that's something that we're working on right now with our colleagues. So just in conclusion, one of the reasons why we're so interested in looking at these different host genetic models is because this isn't the last pandemic we're going to see. And what we really need is to ramp up before we end up with another one so that we have a whole panel of different viral models that we can use to study a new version of a coronavirus. And this is something that we're working on in collaboration with the NIAID to build up this kind of panel for future use when a pandemic hits. So I'll just stop there and say that it usually takes a village to do anything, and in this case it really did. You're seeing all of my friends and colleagues on the top two lines, my collaborators at the NIAID over there on the left in the lower half, and because I also have a lab at Imperial College, several of my colleagues over there, accomplished virologists who are helping with designing and analyzing these interesting mice. So finally, I will just thanks to our real unsung heroes, all those wonderful mice that come in so many different colors and flavors that we're also interested in. And thank you for your attention.